The overall goals of these assays are to quantify monocyte chemotaxis in vitro in real time and to track lymphocyte migration in vivo.
The in vitro method can help answer key questions in the field of immunology, such as what are the effects of serum factors on immune cell chemotaxis. The main advantage of the in vitro technique is that it permits quantitative and multiplexed measurements in real time.
The in vivo method can help answer key questions in the field of immunology, about how specific cell types migrate within a recipient animal. The in vivo technique is particularly useful for the quantitative assessment of the migration of multiple cell types within the same animal.
Before beginning the experiment, label THP1 cells with a fluorescent dye in a 35 mm cell culture dish in complete medium at 37 degrees Celsius for 30 minutes. While the cells are incubating, add 750 microliters of HBSS buffered with 25 millimolar HEPs and 0.1% BSA to the background and positive control wells of a 24-well plate. Add the appropriate concentration of MCP1 in water supplemented with BSA to the experimental and positive control wells, and place porous inserts into each well, taking care that the tabs of the inserts align with the notches of the plate.
It is critical to remove any bubbles from the bottom wells after placing the inserts, as bubbles will interfere with fluorescence measurements.
At the end of the incubation, transfer the labeled cells into a 15 milliliter conical tube for their collection by centrifugation. And visually